Welcome to the Hematology Series, a series of informative and short videos on hematology. In this video we will briefly be covering the neutrophil. Neutrophils are produced in the bone marrow from self-renewing hemopoietic stem cells. This process is called granular poiesis. It is regulated mainly by interleukin-3, granulocyte monocyte colony stimulating factor and granulocyte stimulating factor. Mature neutrophils leave the bone marrow into the peripheral circulation. During neutrophil maturation, myeloblasts are converted into promyelocytes. Promyelocytes then mature into myelocytes and later onto metamyelocytes. The last two stages of neutrophil production include the band neutrophils, which later mature into segmented neutrophils. Neutrophils have a multi-lobed nucleus, and normally it is made up of 3 to 5 lobes, joined by thin strands. The cytoplasm contains purple granules, having microbicidal agents. Other granules contain enzymes such as lysozyme. Cytoplasmic organelles such as the mitochondria and the Golgi apparatus are also present. The average size of neutrophils is 8 micrometers. The main function of neutrophils is to fight pathogens. They migrate to sites of infection, recognize and eliminate microbes by means of phagocytosis. They are defensive against various pathogenic organisms including bacteria, fungi, and protozoa. Using Romanowsky stains, neutrophils can be observed microscopically. Normally, the nucleus appears as purple and the cytoplasm as pale pink. The granules stain pink and purple-blue. However, neutrophils may vary in their morphological appearance, which may indicate an underlying disorder. These are some examples. In toxic granulation, neutrophils have dark staining granules as a response to inflammation. In Alder-Riley anomaly, the granules resemble toxic granulation due to partly degraded mucopolysaccharide deposits. Apoptotic neutrophils involve cell shrinkage, nuclear fragmentation and loss of cytoplasmic granules. This is usually a natural process. In Chediak-Higashi syndrome, neutrophilic granules are reduced and enlarged due to a genetic mutation affecting the lysosomal trafficking regulator gene. In toxic vacuolation, neutrophils have pale patches. This is mainly caused by bacterial infection or granulocyte colony stimulating factor treatment. In pseudo-Chediak-Higashi syndrome, neutrophils appear similar to those of Chediak-Higashi syndrome. These are mainly found in acute leukemia or a myelodysplastic disorder. In Jordan's anomaly, vacuolation is prominent and widespread due to an accumulation of lipid in the cells. In hypogranulation, neutrophils have sparse or lack cytoplasmic granules. These are present in myelodysplasia. A normal segmented neutrophil count is approximately 2.5 to 7.0 times 10 to the power of 9 per liter in the peripheral circulation. They comprise about 60 to 70% of the total white blood cells. Also, a very small number of band neutrophils are normally present. The term neutrophilia indicates a high neutrophil count. This could be factitious due to artifacts during blood sample handling. Primary neutrophilia is due to impaired bone marrow neutrophil production, such as in chronic myeloid leukemia. Secondary neutrophilia is due to inflammation or infection. Neutropenia indicates a low neutrophil count caused by a decreased production with marrow hypoplasia. This can be primary, such as cyclic neutropenia, or secondary due to cytotoxic drugs or disorders such as aplastic anemia and leukemia. Thank you for watching.